So we will be presenting our experiment on the role of the dopamine system in Drosophila sleep. Next slide. For our specific aims for this experiment, aim 1 was mapping the neuroanatomical distribution and projections of dopaminergic neurons in the fruit fly brain. Aim 2 was investigating the influence of dopamine D1-like receptors on fruit fly sleep behaviors. Aim 3 was investigating the influence of dopamine transporter protein on fruit fly sleep behaviors. Next slide. So our specific aim 1 was mapping the neuroanatomical distribution and projections of dopaminergic neurons in the fruit fly brain. More on that in the next slide. Our experiment setup for this was to use GAL4/UAS system in order to visualize all the dopamine neurons in the fruit fly brain. To do this, we use GAL4-8848, which targets all dopamine neurons, and UAS 8515, which is eGFP. So using this, we produced the two images shown below on the slide. The one on the left is less magnified and the one on the right is more magnified. And you can see the individual dopamine neurons glowing green in the blue light and the yellow filter used to image these pictures.
Okay, so next we're going to be talking about specific aims 2 and 3, which were to investigate the influence of dopamine D1-like receptors on fruit fly sleep behaviors and investigating the influence of the dopamine transporter protein DAT on fruit fly sleep behaviors. Next. So in order to study these two systems, we created two separate crosses. Our first cross was done by crossing the GAL4 79260 flies with the UAS 28896 flies. And this created a fruit fly cross that had the expression of the DAT protein silenced. And then we created another fly cross with GAL4 19491 and UAS 41753 to create a cross that allowed us to optogenetically control the D1 receptor expression. And then, as I said before, creating these two crosses allows us to study the relative effects of the silencing of D1 receptors and DAT protein on sleep cycles.
So both of our AIM-2 and AIM-3 flies, along with our control flies, were exposed to the same experimental conditions in order to maintain consistency across our experiment. DAT expression was inhibited via RNAi, and D1 receptor expression was controlled optogenetically with halorhodopsin. As seen in the picture on the right, we used a flashlight held in place above the Drosophila activity monitor to expose all of our flies to yellow light, which activates halorhodopsin and allowed for the inhibition of D1-like receptors. Data was collected over the course of 24 hours with the goal of being able to compare the differences in fly sleep behavior depending on whether the receptors were activated or not.
Okay, so I'm going to talk a little bit about how we analyzed our data. So overall, our approach was to quantify fruit fly sleep behavior by using the activity monitor to record the total counts for each fly over the course of 24 hours. So we recorded 2 trials for each fly line, and we found that previous studies showed the average fruit fly sleep window was around 623 minutes per day, which is around 10 hours. And such a sleep window would be associated with the least amount of movement during the 24-hour period. So we located a 10-hour span within each data set that was associated with the least activity. And that's what we characterized as the fruit fly sleep window. So overall, we quantified the fruit fly's sleep quality or sleep duration by evaluating the total counts during the 10-hour span for both of our crosses and their controls. So for example, flies that moved more during this period would be considered to have lower sleep duration and a lower sleep quality, and vice versa. So the first way that we visualized our data was by comparing the total counts for each cross line versus their 2 controls. And the graphs show both trials as well as the average for all of the lines. And the average value is labeled on all of the graphs. So in both cases, you can see that the total counts during the sleep window was significantly higher for both crosses in comparison to their controls. And that indicated for us, first of all, that our crosses were successful. And second of all, that the genetic manipulation did have an impact on fruit fly sleep. So with this in mind, our overall goal of our experiment was to determine the relative impacts of each aspect of the dopamine system on fruit fly sleep. So in order to do this, we compared the percent differences between each fly cross and then their two controls. So for example, for the DAT protein cross, the controls were the lines 28896 and 79260. And for the D1-like receptors, the two controls were 41753 and 19491. And so for visualization purposes, we formatted the data in scientific notation, and that was just because the percent differences were very large. So the two main conclusions that we drew from the data were, first of all, that both manipulations had a very dramatic impact on fruit fly sleep. And second of all, that the impact of blocking dopamine transport protein caused almost twice the increase in movement compared to blocking D1-like receptor function. And that indicated to us that the dopamine transport protein had more of an influence on Drosophila sleep than the D1-like receptors. And so overall, our conclusions were that we were able to accomplish all of our specific aims. So first of all, we successfully visualized the fruit fly dopamine system using GFP. And we were also able to successfully evaluate the impacts of both D1-like receptors and dopamine transport protein on fruit fly sleep. And we found that the influence of the transport protein was almost twice that of the receptors.
So in every experiment, it's very important to consider potential experimental error. And for our experiment, a significant portion of potential error came from the fact that the environment where we were conducting our research, there were multiple other groups also synchronously running their crosses in the same DAM monitor. So the first thing that really kind of may have affected our data is the changes in settings that other groups sometimes did during the recording period. While we weren't there. So that could have affected our data, as well as the fact that the computer also stops recording after 6 hours following whenever it last goes to sleep. And then there may have been movement of the fly chambers from the other groups who are also inserting and taking out their flies from the monitor. Another potential effect, mostly for the second and third specific aims, is the flashlight battery life did not last the entire time. So the flies may have been exposed to yellow light for a majority, but maybe not the entire time that we were conducting the experiment and for the recording period.
Moving forwards, the results of our experiment are potentially relevant to the treatment of sleep disorders in humans, since there are many similarities between the two, including modulation by the same neurotransmitters and circadian regulation. As a result, fruit flies are widely regarded as a valuable model for both invertebrate and vertebrate sleep. Most widely available treatments for sleep disorders target the serotonin system, yet they have serious side effects for individuals with bipolar disorder, epilepsy, diabetes, and more. Comparatively, dopaminergic treatments are rare and some are in the process of being developed. The results of our experiment show that the ideal target for a dopaminergic sleep treatment would be dopamine transport protein expression, as this was the component of the dopamine system we identified as most influential in modulating sleep. However, the D1-like receptor function also had a strong influence on sleep, so targeting this could also be potentially effective. Although it is important to note that the further research would be needed to confirm these results.
So to finish off, we wanted to talk about some potential targets for future research. The first of which would be to compare the effect of modulations on the serotonin system to modulations in the dopamine system. So right now, as previously mentioned, most of the medical approaches to treating sleep disorders are to target the serotonin system. But to pivot and start targeting the dopamine system, we want to have a head-to-head comparison to see what kind of potential benefits or disadvantages there would be to switching and targeting a different system. And also, we could run a similar experiment, but instead upregulate these different components of the dopamine system. So in this project, we purely downregulated both the DAT expression and the D1 receptor expression. But by upregulating them, it would allow us to examine the effects of increased activity of the dopamine system on sleep, and it could give us a better understanding of what a pharmacological treatment approach would be best suited to focus on. Yay.